Today I'm describing a very interesting topic, and the topic is the fast display technology and update. The fast display technology was first invented in 1985 by George Smith, and he received a Nobel Prize in the field of chemistry in 2018. Basically, what are the phages? Phages are composed of nucleic acid that is surrounded by capsid protein, and the P. George Smith used filamentous phages for the displaying their peptides. That's why mostly for the fast display technology, the filamentous phages are used. What are the basic principle of the fast display technology is the gene of interest that is inserted nearer to the coat protein gene, which encode the coat protein. So it may leads to expression of our gene of interest outside, and the peptide is displayed on the coat protein, which is linked to the coat protein. That's why they that peptide which are displayed to outside that are used to bind a particular type of unknown target to which we can identify the actual peptide sequence of our target. The fast display technology is helpful for the study of protein-protein interaction, protein-peptide interaction, or protein-DNA interaction, and it is very useful technique because of for the development of different vaccine kit and different therapeutic antibodies for the development of diagnostic kits. As I told that the filamentous phages are used and mostly in the filamentous phages, FD phage, F1 phage, and M13 phages are used. The filamentous phages are inserted with a particular type of gene of interest that may leads to whatever the inserted gene inside is getting expressed outside. Means we can match whatever the inside as genotypically that are also expressed outside phenotypically. And in that principle, because of the same genotypic and phenotypic expression, we can measure either the what was the sequence of the DNA or what was the sequence of amino acid peptides outside. Now comes to the structure of M13 phages. These are the filamentous phages which are cylindrical in nature, and the particle size is about 390 nanometer in length and 6.5 nanometer in diameter. As I told that this is composed of a capsid protein and inside is the nucleic acid. The capsid protein is again divided into the major coat proteins and minor coat proteins. Uh, the major coat protein is about 2700 in numbers, and the minor coat proteins are about 3 to 5 in numbers. Minor coat proteins are actually called as P3 protein, P6 protein, P7 protein, and P4 protein, while the major code proteins is only one, that is the P8 protein. So whatever the gene of interest is inserted inside, if it is inserted near to the gene encoding P8 protein, so it may leads to the expression of our peptide outside, which is linked to the P8 protein. If our gene of interest is inserted with the gene encoded protein 3, so the expression of our peptide will be at the which is linked with the P3 protein. The genome size of the M13 is about 6.4 kilobase per inch size, and it is divided into the three different domains. First domain is the replication gene domain. This gene domain is responsible for the translating different enzymes which are responsible for the replication of our Phage particle. Second is the structural genes. This gene portion is responsible for the translating different peptides which are helpful uh, for the synthesis of capsomeres. And these are the assembly genes which are not the structural proteins, but they are useful during the packaging of the phages. The replication of these phages takes place as a rolling circle mechanism, and the gene can be inserted just double to the natural genome. Means we can insert about 12 kilobase pair of size of our gene of interest with the circular genome. Now, as we know, there is too much importance of P8 protein and P3 protein in the fast display technology. That's why I am discussing here what what are the different domains of P3 and what are the actual structure of P8 protein. P3 protein is composed of three main different domains. First is the N1 domain, second is N2 domain, and third is the CT domain. All these domains are linked to each other with the glycine-rich linker. The N1 domain is responsible for translocation of DNA from the phages into the bacteria. N2 domain is responsible for the binding of the bacteriophage to the bacteria with the help of pilus. And the CT domain, that is the C terminal domain, is the integral part of the P3 protein. So how these domains work during the replication of the phages? N1 domain is responsible for the translocation of DNA, so that the N1 domain binds with toll A receptor. This toll A receptor is found intraperiplasmic in the intraperiplasmic region. N2 domain binds with pilus. This is the pilus, and the pilus is the structure of bacteria. This is useful for the conjugation mechanism of bacteria. And this is the C terminal part, which directly binds with the phages, and this is responsible for retracting the phages inside of the bacteria. Finally, after the retraction of these phages inside of the bacteria, the phage release its DNA. That DNA getting directly RF form, any replicative form, that is the double-stranded DNA. First, it was the single-stranded DNA, but when it inserted inside, it will convert it into the double replicative form. There is expression of a different type of the proteins like enzymes which are responsible for the replication. Then some proteins which are assembly proteins which are responsible for the packaging. So here the G5 protein is responsible for the initiation of packaging of the bacteriophage. When the concentration of GP5 protein increases, it decrease or it inhibit the replication mechanism, and it initiate the packaging of bacteriophages, and it also initiate the synthesis of GP8 protein. When the GP8 protein synthesizes, it binds to the peptidoglycan layer of the bacteria, and when the sufficient amount of GP8 protein bound to the peptidoglycan layer, the phage particle releases. Within 6 hours, one phage is getting converted into 10 to the power 10 phages. So the replication is very fast in the bacteriophages, and it is very helpful for replicating and to increasing amount of our protein of interest. Now, this is the structure of the GP8 protein. GP8 protein is helical in the nature, and if you can see that GP8 protein form 20 degree angle from the longitudinal axis, and it is helpful to link or display 2700 types of peptides on the phages because of their inclination. During the fast display technology, about 6 amino acid long amino acid peptide can be linked to the GP8 protein, and about 36 amino acid length of peptide can be linked to the GP3 protein. So this is all about the basic principle and structure of the phages. Now, very important topic is the biopinning of phages, and this is a part of fast display technology. What is the biopinning? Actually, as we know that there are displaying different type of the peptide library outside of the phages, but we have to know the exact affinity of this peptide library to our unknown target. For the knowing the exact target and our peptide binding affinity, the biopenning is most crucial method of the fast display technology. Biopenning is basically the affinity selection method, affinity selection technique method, which can be performed either in the laboratory or in vivo, means either in the animal model. Biopenning is again divided into five different steps. Like first is the library preparation, target binding, washing, elution, and amplification. If we are talking one by one, first is a preparation of peptide. So there is one example with the example of by using example of antibody library preparation. If the animal is suffering from any disease or if the animal is getting vaccinated, there is increased concentration of activated B lymphocytes. During the activation, there are different genes which are rearranged in the B cell, like variable region, then D region, and joining region, etc. So, after the class switching of the antibody, a monoclonal antibody are getting synthesized. That means in the case of B cell, there are different type of gene arrangements are there, and during this gene rearrangement, we can remove the B cells from the donor, and we can extract all these genes. These genes can be incorporated or can be ligated with our uh, filamentous phages, and after the ligation with the our phages, uh, we will get some different type of the expressed antibody outside of the phages. Now these different fragments of antibodies can be displayed and can be used to um, to to interact with our unknown target, or we can say if the target is known, maybe the antibody can be unknown. So either detection of a particular sequence of antibody or either detection of target is needed. The fast display technology is very helpful for detection of either ligand or either receptor or either target or either antibodies. This detection can be visualized either with the help of ELISA, with the help of any fluorescence technique, with the help of any electrochemical technique. So the ligation is very crucial, very important, and it is performed routinely in the laboratory with the help of blue white screening, and the white colonies will be picked up for the correct insertion of our gene of interest to the nucleic acid or to the vector of phages. And these are the different type of the fragments of antibody which are expressed on phages during the phage display technology or during the preparation of antibodies. Actually, this is the main picture of the immunoglobulin. As we know, the immunoglobulin is composed of Fc region and Fab region. The Fc region is composed of heavy chains, while the Fab region is composed of heavy and light chains. But as we know that only the variable portion of heavy and light chain is responsible for the binding of a particular epitope, and it contain the complementary determinating regions which directly bind to the sequences of amino acids of the epitope. So there is no need to display the whole immunoglobulin, because the whole immunoglobulin is very heavy molecule, and the expression of whole heavy molecule is not possible in the laboratory. It is very cumbersome, and uh, it is very time consuming. It is very laborious, and it is worthless. That's why only the complementary determinating region can be amplified in the phages, and we are getting different type of the fragments of immunoglobulins. The next step of the biopenning is the target binding. The target binding can be done either in vivo technique or by in vitro technique. With the help of in vitro technique, by using different microtiter plate or by using any cell culture plate, the different peptide library of the phages can be used to bind with a particular target. Whatever the strongest affinity of the binding of particular phage to particular target is getting eluted and which are used further for the fast display technology. If we are talking about the in vivo method, different animal models can be used, like here the mouse model is used in this picture. As illustrated in this picture, the phages are injected into the tail vein, and these phages having different peptide library which circulate in the body and binds with different organs. If uh, we want to detect any tumor in the animal, so as we know that when there is tumor formation, different tumor homing receptor sorry different tumor homing peptides are released from the tumor, and these peptides are found on uh, tumor surface or in the blood circulation, and that will be detected by the fast display peptide libraries. So likewise, if these peptides bind to the brain tissue, to the heart tissue, to the lung tissue, we can also detect the different uh, structure, different surface area, different cells of the different tissue. The detection of this binding can be done with the basic principles of transducer with the help of electrochemical, fluorescence, absorbance, transmittance principles, and the signal will be generated. These signals will be collected by monitor, by the transducer, and the data will be read out. This data is very important to identify the correct target ligand binding. The next step of biopenning is washing. Washing is performed for removal of loose target ligand binding, and this can be performed by using PBS that contain 2% skimmed milk powder or 0.1% twin 20. The next step will be the elution of biopenning. In this step, those phages which has a strong binding affinity with our target, that will be eluted. The elution will be performed by using higher concentration of urea or low pH value. अ, इसमें एक स्लाइड बैक हो गई है. तो what is the application now? Comes to the application of fast display technology. So there are basically applications of the fast display technology are: first is epitope mapping, second is epitope mimicking, third is identification of new receptor and ligands, fourth is the organ targeting, and fifth one is the um preparation of vaccine. So all the application uh, are only useful for the development of diagnostic reagent, for the development of therapeutic antibodies, and for the development of effective vaccines. Here, first example for development of diagnostic kit and vaccine kit by using fast display technology, as Chitre et al. Reported that why the FMD vaccine get failed, because there is a change in the proline to serine and leucine to phenylalanine at the region of 149 and 150 of number of amino acid at the VP1 protein. VP1 protein is the part of capsid protein of the FMD virus. But here, this substitution in the epitope can be detected with the help of short chain fragment, variable fragment of chicken immunoglobulin antibodies that can be used for the detection of epitope change. So that is a good example of epitope mapping. The detection of particular linear sequences of the peptide or quaterly or tertiary structure of peptide can be identified with the help of Phage displayed peptides. These peptides can detect any change in the epitope. It is very helpful for the diagnose particular vaccine failure, can be diagnosed for uh, preparation of new vaccine by using this anti-idiotypic vaccine. And second is the epitope mimicking. जैसा मैंने भी आपको पहले बताया, the epitope mimicking is responsible for mimicking particular type of a receptor, particular type of a ligand, and it is very helpful by the fast display technology for the treatment of cancer, for the anti-allergic treatment, and for the contraceptive vaccine preparation. Next is the fast display based vaccine. The fast display based vaccine can also be helpful to protect individuals by inserting its DNA inside of the individual. For the better vaccine quality, it is needed that this gene can be inserted with eukaryotic acid, because eukaryotic acid having a strong promoter at 3' region and 5' region. Due to the strong promoter, the strong expression of a particular peptide is getting outside, and these peptides will be acts as a vaccine carrier. Vaccine candidate. The phages acts as a carrier here, and it carries the DNA of our gene of interest. The Due to the DNA vaccine and due to the fast display peptide vaccines, they are responsible for activation of humoral immunity and cell-mediated immunity. With the activation of humoral immunity, uh, they produce they activate different type of the B cell, and these B cells getting activated, and there is formation of different monoclonal antibodies, and when the huge quantity of monoclonal antibodies is there, it will directly bind to the target, leads to the maybe phagocytosis, maybe agglutination, like that, and there is destruction of the particular target. If we are talking about the cell mediated immunity, the phages provide different peptides. These peptides uh, forming vesicles inside, and there is formation of endogenous pathway of antigen. These antigen are getting expressed outside with the help of MSC1 and MSC2 molecule. These both molecules are responsible for activation of cell-mediated T lymphocytes, and there is a release of interleukin 2, interleukin 4, then interferon gamma, tumor necrotic factor alpha, perforins, granzymes, and all these are responsible for um, activation of PD cascade, यानी कि programmed cell death activation, and the Caspar's mechanism leads to the death of a particular target cell. This is another example of vaccine. Here, Yong Cheng et al. Uh, reported that a particular peptide VP2 protein, this protein is displayed outside on the phages by using a particular nucleic acid insertion into the phage, and this display peptide when inserted, when given to the chickens, and then after 4 weeks of the first primary injection, they are challenged with actual infectious basal disease virus. These chickens are protected from this virus, and there were no any mortality. This is a good example of contraceptive vaccine development with the help of fast display technology. Samoilova et al. In 2012 reported that the anti-zona pellucida ligand, which is responsible for binding to zona pellucida 3 receptor in case of wild pig, lead to acts as a contraceptive vaccine because it is anti-spermatic in the nature. What he did, he performed zona pellucida binding peptide screening, and he used different animals because he knows that he knew that the zona pellucida of cat, dog, cow, and pig are very similar. So he performed a peptide selection by using negative selection technique. First, he put the peptides and with the phages into the cat zona pellucida mixture. Then he removed these peptides, whatever which are not bound. Then he further added these peptides into the dog zona pellucida proteins. Then he further removed those peptides which were unable to bound. Then he again put these fast display peptides into the cow zona pellucida reaction. Then those peptides which were unable to bound, uh, they were washed away, and again those remaining peptides were bound to the pig zona pellucida uh, peptide mixture. Leads to the highest binding peptides were uh, found, and they were recovered finally, and leads to use of these peptides results uh, contraceptive vaccine preparation. This is another example of diagnosis of H5N1 avian flu virus with the help of short chain fragment variable immunoglobulins by using fast display technology by Wu et al. This is another example of detection of Pasteurella multocida by using uh, fast display based method, um, and this was performed in the Chaudhary Swarn Kumar Himachal Pradesh Krishi Vishwavidyalay, Palampur, Himachal Pradesh. There is another example of in vivo diagnosis, so it is very important. There is a diagnosis of adenoma in the colon were done with the help of fluorescent based fast display peptide. They were bound to the adenoma of the colon, leads to uh, diagnosis where possible. In case of the therapeutic response of the fast display for the treatment of follicular lymphoma, it was approved by the FDA. The drug was rituximab, and it was responsible for binding with the CD20 large loop molecule. This CD20 receptor is not found in the mature B cell, but when the B cell is getting converted into the cancerous cell, the expression of CD20 is continue occur, leads to production of follicular lymphoma or myeloma cell. And the rituximab is the fast display based peptide which has anti-idiotypic in nature, and that binds with the large loop of the CD20, leads to therapeutic response against the follicular lymphoma. These are the different type of the peptides which can bind to different organs, and there is leads to different cytotoxic chemotherapeutic response can be best seen in case of the fast display technology. These are different type of approved USDA approved drugs, and these are basically the monoclonal antibodies which are used for the treatment of canine arthritis, canine lymphoma, canine atopic dermatitis, and canine parvovirus infection, respectively. These are the few antibody fragments which are responsible for the treatment in the humans of the disease of rheumatoid arthritis, systemic lupus erythematosus, acute lymphoblastic leukemia, then neovascular age-related muscular degeneration, bladder cancer, and anthrax infection. And these all are approved in the England and America. Now there are too many work, and so much work is going on in the veterinary science for not only diagnosis but prophylaxis and therapeutic response of the fast displayed peptide in the different animal species. Finally, the conclusion are the fast display technology is a valuable tool in biomedical application which offers rapid, efficient, and relatively inexpensive methods. Fast display technology investigate protein-protein interactions, receptor binding sites, epitopes, then mimotopes, functional and accessible site from antigens. Fast display technology improves diagnostic methods, design potential therapeutic agents, and vaccines. Thank you.